Hey, yeah, thanks for giving me the opportunity to talk about this project here. I will talk about metabolic microbiome-host interaction in IBD, and we do that in the medical systems biology approach in Kiel using mainly metabolic modeling. And the main idea was to find more or less the singer and less of this, not the singer, but the song of the microbiome, which is played during inflammation in IBD. And we use metabolic modeling to, on the one hand, try to characterize the metabolism in IBD on the host side, but also on the microbiome side, and then associate these metabolic changes and functions we observe to associate that then to inflammation phenotypes. And also response and remission. And using these models, we can also try to identify interventions, dietary interventions, which could be used as a therapeutic option in the future. So we did that in a northern German cohort of IBD patients, which have been sampled longitudinal and have been treated with different biologicals. So it's an anti-TNF alpha and an anti-IL-6, for those of you who are interested. And we sampled colon biopsies, whole blood, and feces. And we did transcriptomic sequencing, metabolomics, and 16S sequencing of these samples. Then we used, so we start with the host metabolism. So what we do is here, we use the transcriptome to infer the expression of enzymes which we know perform certain metabolic functions in a metabolic network. And then we also know the metabolic network of humans, and then we try to estimate which part of the metabolism is active in the given tissue at the given time of that sample. And we reconstruct from that context-specific metabolic models. And we can use these models to also infer the flux of metabolites through the metabolism, and then associate that back to inflammation. And we did that here, and if you do that, you end up with a whole bunch of different metabolic functions which are associated to inflammation. And just to give you an overview, I will show a lot of these plots. Each dot here is basically a reaction in a pathway in the metabolic one, and you see the estimate for a linear mixed model here, and everything above 0 means it's high in inflammation, below 0 means low in inflammation. So that actually just tells you now that there is a lot of metabolic pathways involved, and most of them are downregulated. So we see a loss of metabolic functions with inflammation, and that the blood is more strongly affected here. However, if you look a little bit closer, you can make out some very interesting patterns. For example, if you look at the tryptophan metabolism, which is downregulated in both tissues, NAD metabolism, which is like central to the phospholytic oxidation, and also fatty acid oxidation, which has been described before. Then we ask, are there certain metabolites which are predominantly affected by these reactions? So are they predominantly used by this reaction? And again, we find very prominent across both tissues, blood and biopsies here, these CoA associates, so fatty acid oxidation, and again, energy metabolism, FAD and NAD here. Then we used our 16S data and did basically the same for the microbiome. So we used the 16S, mapped them to reference genomes, and from that inferred the enzymatic repertoire of these microbes, and then reconstructed metabolic models for each of the microbes. Then we put them back together in microbial communities and can infer with dietary information, can infer the metabolic flux through the microbiome, so to say. And if we do that and associate that to IBD disease activity scores, again, we see basically a downregulation of certain metabolic functions in the microbiome with inflammation. And again, we see super interesting things. So some things are very well known. So short-chain fatty acid production is downregulated. Also fermentation here and the starch degradation. So we see basically less fermentation here. On the other hand, we also see that energy metabolism is down-regulated or producing energy metabolites like flavine, so FAD precursor, and NAD de novo synthesis from aspartate, which is specific to the microbes. Finally, we used our metabolomics analysis from the serum metabolomics. So we did targeted metabolomics here. And what you first of all observe is that you see an increase in fatty acid and sphingolipid levels in the serum, while you see a downregulation of phosphatidylcholines. And very much interesting, again, we see tryptophan. And why do I always talk about tryptophan? Because tryptophan is basically one of the major components to produce NAD. So if we bring these results all together, and this is just one major part what we observed here, is that in blood samples we see that there is a major downregulation of, on the one hand, NAD recycling, but also de novo synthesis, either from tryptophan, so you need quinolonic acid as a precursor for NAD. And tryptophan is one way to produce this quinolonic acid in humans, but you also see a downregulation of quinolonic acid production by the bacterial metabolism. So the bacteria will contribute less NAD to the IBD patients. This is slightly different in the gut tissue itself. There we see more NAD recycling. But most importantly, we see one reaction here, which is producing methylnicotinamide. And this methylnicotinamide is actually a dump for NAD. And it's associated to inflammation, that's known, but we actually do not really know what it does. Yet it will draw nicotinamide from the available pool for NAD production. And then the same we see here, bacterial metabolism is not contributing to that. Finally, we wanted to know where the tryptophan of the serum is basically going, and where is it? Yeah, so why is it not contributing to the NAD levels anymore? And if we look at the tryptophan degradation pathway, there are three major ones, but especially in the gut tissue, we see an activation of the quinolinate pathway, which actually would lead to quinolinate here, this NAD precursor, yet the metabolism is exactly at that step is blocked, and it will rather lead to picolinate or acetyl-CoAs here. So we see that the gut tissue, not the blood, is actually contributing to the loss of tryptophan levels in the blood. Finally, we wanted to see whether we can alleviate these metabolic functions in the microbes. And what we did is we used our metabolic models and simulated increase or decrease of certain dietary metabolites and see how that affects the production of different metabolites in the microbiome. And if you look real closely, we actually find a way to increase nicotinamide production in the microbiomes from the IBD patients by removing lactose and sucrose. And this is something which has been described in the literature already before, that if you reduce carbohydrate diet in IBD patients, that this is beneficial, yet we didn't really know why. So this might be one explanation for that. So to sum that up, what I just told you is that we see a decreased energy metabolism that is most likely causing disturbances in almost all metabolic pathways we see across IBD. And then we see that NAD synthesis and regulation is massively disturbed. We see a decreased input from the tryptophan degradation in the gut. We see decreased de novo production by the microbiome. And yet we see some possible interventions for NAD production in the microbiome. With that, I would like to thank my collaborators and my working group, my funding buddies, and you for your attention.
Thank you. Thank you. That was remarkable. We have time for a quick question, if anybody has one. Otherwise, I shall ask one. I'm curious, one step of the pathway, you're using 16S data and then you're mapping it to MAGs, but are they MAGs from the samples or are they just in reference databases?
This is basically a reference database from a very similar sample from MAGs, from also IBD patients, and it's spanning worldwide samples, but also includes IBD patients.
So you're convinced they accurately describe the bacterial genomes? Because that will
Probably
then
not. Probably not. Probably we will. Well, we at least hope that we are close enough to the metabolism. I mean, what we see in the metabolic prediction, what we do there, that very well corresponds to what we see in other literature, and so known phenomena are well described with our approach. So we're quite confident that we're at least not completely model out of thin air.
Oh, super. It's very good. Well, thank you. We'll move on then. So next up,